I would now like to present the next lightning talk, the next and last one, by Velia Veronica Rangel Ramirez on genotyping of interferon gamma, polymorphism using T-ARMS-PCR technique in Mexican population infected with SARS-CoV-2. Veronica?
Yeah, I'm here. Thank you.
Welcome.
You can see me. Thank you. Okay, I'm going to share my screen. Can you see my presentation?
Not, yes, it's starting to share.
Okay, can you see it now?
Not yet. Yes, we can see it now.
Okay, thank you. Well, thank you.
Okay, we only see, is it just one slide or you have?
Yeah, I'm going to keep sharing some image.
Okay, go ahead.
Okay, well, thank you for the consideration of my work. The name of my project is Genotyping of Interferon Gamma in the position 874 using T-ARMS-PCF technique, as you have said. When we talk about SARS-CoV-2, we can talk about other things like therapy, like clinical features, like prognosis, the virology, etc. However, we know that this virus is characterized by a respiratory syndrome with a variable degree of severity. In this regard, the immune system are very important to fight this infection. So we know that there are many cells that contribute to this fight. Between them, there are T cells, B cells, innate immunity like natural killer cells, etc. However, we also know that very important for the action of these cells are the cytokines. So cytokines are small proteins that can help to communicate between cells, and among these cytokines there are interferon gamma. Yeah, well, there are different types of interferons, but we in this talk we're going to talk about the interferon gamma. In this regard, interferon gamma has a role also with SARS-CoV. So there has been some association between polymorphisms with this interferon, which function is to stop the viral it has a function in antiviral activity. So the objective of the aim of my study was to investigate the role of the polymorphisms of this interferon gamma at the position I told you about in the context of the susceptibility of Mexican individuals infected with SARS-CoV-2. For this, we use a technique to identify SNP polymorphisms, which is called T-ARMS-PCR. So in this case, what we did was to see in a gel stained with ethylium bromide, like a normal PCR, SCP, endpoint PCR. And what we did was to find out if we saw these two alleles, A or T, of these polymorphisms in a gel. So this there was no need to see or to use sequencing for identifying the different alleles. And once we did that with the primers, what we did was to compare between the two groups that we have. We have the SARS-CoV-2 group and we have the control group. I know that my number of individuals are small, and for these studies we also need to make it bigger, but for now we have 48 SARS-CoV-2 positive individuals and 24 controls. I'm sorry, 28 and 24. So as you can see in the first table, in the top, you can see that we separate in the genotype AE or TT, and we don't see any difference between the genotype or the allele A or T for the two groups until now. But I told you the number of individuals are still small. We also, what we did was to compare between the symptoms with the three genotypes that we have, like fever, like dry cough, like headache, dyspnea, chest pain, myalgia, etc. And we still don't see any difference between the different genotypes. We also try to analyze the comorbidities of these people, but we don't find until now any difference. What is the importance in this case? Well, if some allele is more present than the other one, is going to change the expression of the interferon. So it's better for the population one genotype or the other. In this context, it has been observed that the polymorphisms has some better prognosis in hepatitis B or tuberculosis. So for now, the conclusion of my work is there is no difference until now between the two groups, and we hope to make it bigger, like 300 people for each group, so we can see if there is some relationship between this and the disease. So, well, I just thank you for to the University of my state of San Luis Potosí in Mexico, and the work, where the place of my work, which is called Center of Bioscience. Thank you.
Thank you so much, Veronica. That was quite an interesting topic. Interferon gamma role in COVID has really been something significant and has really been a focus of attention for a long time, and we hope that there's good yield in understanding it. I would like to ask if Eleanor, who's quite known in this field, if she has any question for Veronica or any particular comment on such work.
No, I think that was very succinct, Veronica. Thank you very much. We followed it all. As you said, we'll wait eagerly as you increase the numbers that you can follow, but a great poster presentation. Thank you.
Thank you.